So I want to take a minute and talk about heme. So heme is part of this learning unit because heme has nitrogen atoms, and so it's built from some amino acid sort of building blocks. But when we think about heme, we actually see heme in a lot of different places. So when you think about heme, I want you to think about a structure like that. Big picture, some things that I hope you take from this course is when you see certain molecular structures that you recognize, oh, that's related to a steroid. Oh, this is a protein. Oh, that's a nucleic acid. That's a heme derivative, right? I want you to be able to recognize these different classes of molecules. So let's just brainstorm for a second here where we see and have found heme throughout our study in this course. Well, we've seen heme in red blood cells, right? It's an important component of hemoglobin. We've seen it in muscle tissue, right? An important component of myoglobin. We've actually seen it in the electron transport chain. We've got lots of cytochromes, but specifically we've got cytochrome C as a hydrophilic sort of mobile carrier of electrons, right? We talked about cytochromes P450. That's found in liver detoxification. And we highlighted that when you talk about Tylenol overdose, one of the reasons that Tylenol overdose is problematic is because it sort of taps out your cytochromes P450 detoxification pathway. One of the last things we talked about is it is in a modified form found in chlorophyll. So it looks a little bit different, and importantly, it is going to have that iron sort of replaced by a magnesium metal ion in the middle. So we've seen a lot with heme. I'm not going to go through the details for how we synthesize heme, but if we think about what a recipe is for heme, one of the things that I want to sort out here and highlight is the fact that we have carbon atoms that we need to have and nitrogen atoms that we need to have. So fundamentally, when we're thinking about carbon atoms, a great place to siphon building blocks off of is the citric acid cycle. So the citric acid cycle is going to be a great source of carbon atoms. And then amino acids are going to be a great source for nitrogen atoms. So just as a theme, as we're building molecules, think about where we can get those building blocks from. Lots of citric acid cycle intermediates can be siphoned off. Remember, we talked about the citric acid cycle as having both cataplerotic and anaplerotic sort of directions, sort of feeding into and feeding out of in terms of breaking things down or using those intermediates, those metabolites, to build other molecules. So that's all I'm going to talk about in terms of how we build heme. One thing I do want to spend a little bit of time talking about is actually how we break down and degrade heme, because degrading heme actually has an interesting and very colorful, literally, story. Okay, so first let's sort of talk about when and why we'd want to discard or break down heme. Well, maybe we've got old red blood cells that we need to discard. So remembering that the life cycle of a red blood cell is about 90 to 120 days. Okay, so maybe we've got some old red blood cells that we're sort of disposing of. Maybe we have some bruising. You spell bruising right. Some bruising that we have in a tissue, right? So we've got heme that we need to sort of get rid of and break down there. And an inability to discard heme. So again, if this is malfunctioning, if we've pinched our pathway somewhere, that ultimately can lead to jaundice. And we're going to highlight how that happens. And this is particularly problematic with people who have liver disease or infants. And so we'll talk about how and why that is. So the colorful story I'd like to share with you here is looking at sort of the structures of heme as it starts out and how it changes in color as it actually gets broken down. Okay, so here is the structure of heme. And heme, when it starts, is red in color, right? We talked about that's the reason that hemoglobin is red in color is because that iron that's bound inside that heme macrocycle is going to generate red in color, specifically when it is bound to oxygen, right? We talked about the difference between oxygenated and deoxygenated hemoglobin. So as we go through this degradative process, and I'm not going to go through the details here, but you can see sort of a first step is we've got a ring opening that we have here, and it generates something called biliverdin. So biliverdin is going to be green in color, as the name biliverd, verdin refers to it as being green in color. So biliverdin is green. Okay, and then we have it made into bilirubin. And so bilirubin maybe is something that you've heard about before. Okay, this is going to be the piece that is problematic for individuals who have jaundice. We have a buildup of bilirubin. And then once you have the ability to get rid of bilirubin, it can be made into urobilin or stercobilin, which are compounds here. Their color is really reflected in what color they appear as. That urobilin is sort of yellow in color. This is excreted in the kidneys. Okay, and this is going to be responsible or partly responsible for the color that we see in urine. We highlighted in class that sometimes your urine might be colored based on things that you've eaten, particularly B vitamins and a multivitamin can actually make your urine almost fluorescent yellow in color. And then if it is something that is disposed of in the large intestine, so in your fecal material, this helps contribute to the brown color in stool. So lots of things are going to be coloring your waste products. Want to highlight here that we have sort of an important component of your microbiome that's sort of contributing to what we see here. So just kind of highlighting some practical aspects of what we have here. If you look at sort of the degradation of a bruise, I'm sure everybody has had a bruise. When you first get that bruise, maybe it doesn't even show up as a bruise at first, but then it sort of starts becoming that purpley reddish color. That's when we're starting to sort of have that heme break down. But as that heme sort of gets broken down, we get to the different colors of a bruise, which basically are going through the greens and the yellows that we have as that heme is broken down, right? So just highlighting here, we've got waste heme products are eliminated in both urine and stool, right? And here's an individual that's pretty severely jaundiced. So if you have an inability to sort of get rid of bilirubin, if this part of the pathway kind of eliminating bilirubin is pinched, you're going to accumulate bilirubin. Very insoluble sort of species. So what happens is your body tries to find a way to get rid of it, and it deposits it in your skin. So you're going to notice it as a yellowing of your skin. Particularly, you'll notice at first yellowing of the eyes, and that's just because, you know, your eyes are white in color, so you see that discoloration fairly easily. Infants sort of, have a problem with disposing of heme, and they have a buildup of bilirubin, especially if they're not urinating, because if you're not able to excrete this out in your urine or your feces, you can have an accumulation. So particularly infants that are breastfed, and maybe as mother's milk is taking a little while to come in, if they are not urinating frequently, they're not able to get rid of that bilirubin, and infants can become jaundiced fairly quickly. So interestingly enough, one way that you can sort of break down this bilirubin that appears in the skin is with light therapy. So sometimes they will have light therapy that they can use, particularly with infants. Somebody who has sort of a systemic sort of malfunctioning liver problem, that's not going to be a solution there. But oftentimes light therapy can be a way to jumpstart bilirubin sort of elimination in infants that are just sort of waiting for that process to get started. So again, heme breakdown, pretty interesting story. And again, we've got a pretty colorful story to think about there.